So with that, let me introduce Lillian Su. Lillian is a senior staff physician in the Division of Medical Oncology and Hematology at Princess Margaret Hospital. She's also professor of medicine at the University of Toronto. She directs the Phase 1 program and is co-director of the drug development program at Princess Margaret. She's won a number of awards, one of which is the Oncology Drug Development Award from the NCI, the US NCI in 2010. Near and dear to my heart, and also Marge. She's been on the faculty of both the Vail course as well as the FLIMS course, which is now the Zeiss course over in Europe. One of the most rewarding things in our careers is teaching the next generation of investigators how to conduct investigations. She's on the editorial boards of JCO, European Journal of Cancer, and Cancer Discovery. And most importantly, she has served on the board of ASCO several years ago and is currently on the board of the AACR. So truly, there are no boundaries in the people that Lillian works with and the organizations she works with, because the enemy is cancer. And to do that, we have to build teams. So Lillian, thank you for joining us.
Thank you, Lee. I want to thank Lee and the organizers for the opportunity to speak in this plenary session. So several months ago when Lee emailed me and he asked me what I want to talk about, so I said precision medicine. He goes, no, that's not going to work. You need to talk about something a lot more challenging. We are now really a decade from precision medicine. Where do we go from here? I hope I don't disappoint you, Lee. This talk is really some thoughts that I have put together where we should, and perhaps all of us, think about where to go from here after a decade of precision oncology. I do have disclosures, so I clearly missed the deadline. Sorry about that. Here they are. I want to start with this tweet. I learned how to Twitter this year, so it's really interesting for me. And I've been following Eric Topo, and this tweet really caught my eye, and I know it has been retweeted quite a few times by many people. But I think it really does encapsulate a lot of what's happening in the world of precision oncology. I mean, clearly we now know everything is individualized, that we no longer are really treating only the population. We need to personalize and understand individual patients' genomic and immunotherapeutic vulnerabilities. We no longer really doing one-off doctor's office. It's really real-world data, real-time assessments. As much as the doctors are taking charge, patients are very much in the decision-making process. Our medical records is not just a piece of paper in our records. Patients have access, and there is definitely active interaction. The information is owned by the rightful owner, whoever we feel that should be. I think next-generation sequencing and other technologies are becoming much more affordable. And really, the view is no longer limited by data. It's panoramic. We're looking at the whole dimension of the patient, the tumor, the immune system, and the microenvironment. So I'm going to start with that. So a few words about where we have taken precision medicine in Princess Margaret. I'm fortunate enough to have access to a good Phase 1 program and also linkages to the genomics program and the tumor immunotherapy program. So what we wanted to do really is to bring these into one core vision of genomics and immunotherapeutics and drug therapeutics, precision oncology. And I think with that, we have been trying to make sure that every project that we launch in the precision medicine enterprise is really taking advantage and leveraging on all these expertise. Obviously, also working with our radiation oncologists, surgical oncologists, as well as medical oncology, and other colleagues such as pathology. So let me move now to where we have been. I think in the last many years, we're all very familiar with large molecular profiling programs around the world, Europe, America, as well as Asia. And these are just some examples of molecular profiling initiatives and genotype matching activities that have come to fruition and published. So we are aware that, for example, the French group in the Moscato study just published in Cancer Discovery of their over 1,000 patients, 19% of those patients were matched to genotype matching clinical trials. The The Curie, when you look at the Shiva trial, that was the first randomized study of this precision oncology strategy, about 6% of the patients were matched to specific aberrations. The BCCA Vancouver group, using whole genome sequencing, in their first published case series, matched 1 of the 100 patients. MD Anderson, 4%. Our own series, about 5%. Obviously, what's catching the eye is that these are not large numbers. These are all less than about 20% at the max, on average about 5 to 10%, using different technologies, be it next-generation sequencing, or in some cases, some addition of some transcriptomics, some using fresh biopsies and some using archive FFPE samples. I think the obvious question is, was precision medicine really achieving its goal to make everyone benefit? I think obviously the answer at this point is we don't benefit everybody. And obviously in some patients that we do benefit, we are seeing some long-term survivors and some long-term disease control. What we have tried to do in our impact and compact study is to look at patients in a obviously non-randomized fashion of those who were genotype match versus those who went on a clinical trial that was not genotype match, whether there's a difference in terms of objective response, and we saw a difference between 19 versus 9%. Totally granted that this is a non-comparison in terms of randomization, but I think we're all trying to really get some metric that matching is beneficial. Many have argued that perhaps it's really not a populational assessment that we should be making. It should be really the individual drug, individual target, and patients that benefit. Even if you benefit 1 out of 100 that has a long-term disease control, you have achieved the goal of precision medicine. The Shiva trial I have mentioned by Christophe Lachonneau, one of actually our fellows at Princess Margaret many years ago, who returned to design this trial back home in France, and really trying to look at, in a randomized way, matching patients to specific drugs, whether it can achieve a proof of concept. There are some caveats and limitations to the Shiva trial. I won't go into exact details, and I think many of you are very familiar with the Shiva trial, but nevertheless, you can see that the curves are overlapping. So as a result of that, in the last couple of years, editorials, including Ian Tannick from my own institution, has written in the New England Journal of Medicine, saying perhaps there are limits to personalized medicine, and perhaps this whole precision oncology is an illusion. And I guess my question today is, how do we change this illusion to something that is a lot more concrete, and how do we really take it to the next chapter? So I think at this point it's clear to us that not every finding from NGS or even from other technology is druggable. There's a significant proportion of patients, even after whole genome sequencing or exome sequencing, or even other technologies, that we know we do not identify an actionable target. I think what is in the white space in this circle is really our challenge for the next decade. And what we have done in our group is really trying to understand how we can best use the information that has been generated to take it to the next step, to find more actionable targets. So this is an exercise where we have done to look at data from TCGA, over 2,000 patients, using our 500 gene panel, a homebrew panel, which is really no different from some of the other panels by commercial vendors or in large academic institutions. 400, 500 genes is quite common. And when we try to look at different company pipelines, this is just one example of a company that has these various targets. We're trying to see really what kind of percentages in the best scenario would have an actionable aberration. And in that you can see even in the bottom, we will even look at high mutational burden as a potential match that we can use this to find specific agents for these patients, assuming that in all best scenario that they have a good enough performance status, et cetera, to go on such a study. As you can see, in the best scenario, we think 80% is possible, but at the end of the day, if you remember, we only match about 5 to 6 patients, or 6% of the patients. And obviously, there are challenges that make that attrition to occur. The other thing we're trying to do is really, again, interrogate various sorts of data, including TCGA, really looking at immune targets that have been published, see how we can really understand which immune targets may be relevant in various patients. And we're going through this kind of exercise now. I would argue that many immune oncology combinations, and I know Mario will give you a lot better insight, at this point are really empiric driven and not really based on scientific evidence that those targets are relevant in specific tumors in the microenvironment. So I think, again, another exercise that we're trying to do is to really try and understand beyond the simple point mutation that is clearly druggable, what else can we use the data to drive to the decision of therapy? Many reasons obviously led to the point that despite 80, 90% we think are potentially druggable, we end up being less than 10% in many cases. And these are lessons I think we have learned in the last decade of precision medicine. Clearly, by opening more clinical trials, we can capture more patients, and we are seeing a large variety of genomic-based clinical trials, baskets and umbrellas opening fairly rapidly, and many of them in cooperative groups such as SWOG. And I think physicians and patients are much more intelligent now in this area, much more savvy in terms of finding out trials. It's not uncommon that we get an email every day from patients or from other physicians saying, I have a patient with this particular mutation. Do you have a trial open for them? And having resources to come to different parts of the world to actually enroll in a trial is not an uncommon phenomenon. So I think there are many barriers we can break, and these are just some of them that I have listed here. What are the gaps? I think it's clear that not all of the genome is druggable, and even if the genome that is druggable, we may not necessarily have a suitable clinical trial that is open. And I think it's obviously that most of us can find rare mutations in our own institution, but we don't have the power really to test, other than N of 1 scenario, such kind of matching activity. So it's important that we work together, and we're going to hear from Dr. Foti in terms of the whole power of team science. By working together, perhaps it is much more feasible than to assess these rare variants. And obviously, like others, we're looking at other omics, we're looking at functional testing, including genomic, functional genomics, et cetera. And then obviously the challenge is really still looking at heterogeneity and evolution. Most of the studies that are currently in the therapeutic arena still looks at one sample and not dynamic evolution of multiple samples that clearly is important. And obviously we need to learn to data share. So I think one element that we have learned to be very important is that all our genomic or immunophenotyping activities in our institution have a data sharing clause now, because without that, we feel that we will not be able to do so with clear ethics approval. So we have included this phrase almost generically in our consent to do that. We have started to look at various initiatives that I will highlight to you that perhaps is really our own venture into the next generation of beyond precision oncology. We have grouped together with various other centers in our province to really help to get the data together so we have a larger set of samples that we can potentially then run clinical trials for rare variants. So this is not sequencing for them, but really using their own sequencing facilities and personnel to collect the data together, to learn together. And what we are also asking in this particular study called Octane is that each patient also donates an extra sample, and we're using archive sample in this situation, and allows us to do much deeper dive in a translational genomics laboratory. We have the privilege to also go back to ask these patients for a fresh biopsy, but it would have to be under a separate protocol. But I think this is really allowing us, again, to work together to find the rare variants that a single institution cannot possibly identify or study in great detail. This is a study that we're opening through the CCTG, or the Canadian Cooperative Group. Actually, the Cooperative Group meeting is happening at the same time as SWOG. And myself and Dan Renouf, a young investigator from Vancouver, is the co-PI for this study. This is actually very similar to ASCO's taper, where we are getting approved drugs from various pharmaceutical companies to really test for off-label indications in patients who have had molecular profiling that we want to see whether the genomic matching activity is going to be beneficial. And these are the 4 groups across Canada, Vancouver, Ontario, Quebec, as well as pediatrics. We're pulling our data together, and then what we're trying to do is to find drugs from various companies with approved indications to test them in that off-label setting. And we are going to share our data with ASCO's taper as well as the Netherlands activities, which is called DRUB. And it's going to be about 30 baskets of these trials open. Each one will be started based on a particular histology and a particular gene, for example, lung cancer, BRAF, and all different variants, whether they are really known to be deleterious or even of unknown variants. But we ask each group that contribute patients also share the genomic data so we can really understand at a much deeper level what the different variants and different disease histology means in the context of drug matching. So this study is actually hopefully opening in the next couple of months through the NCI Canada. We are starting with single agents, but I think many of us are already thinking single agent is not the way to go. We're thinking of how to take capture to the next level already, even though it itself has not opened. And obviously combination is going to be key. But the possibility of opening combination study is so large that I think there has to be some sort of smart way to do that. And many of you are familiar with adaptive strategies where we're really trying to let the winners win and drop out the arms that are clearly not winning. And I think we're putting our heads together now, thinking of how best to design the next study where we're going to evaluate combination strategies, whether it is targeted immuno or cytotoxic chemotherapy in doublets or triplets. I mentioned about taper as well as the DRUB, which is really similar activities of testing approved agents. Again, we plan to data share. The other activity that we're doing also is to work with Vancouver, which is one of our good partners in Canada, where we are looking at creating the data portal that we can allow us to share clinical as well as genomic data. So one of the study is really a very simple study, only patients with colorectal cancer who have liver metastases that are resectable. We plan to do whole genome sequencing in BC, epigenetic profiling in Toronto, circulating DNA in both centers, and really trying to figure out how we can share and discuss the findings across the country, only 5 hours apart by airplane, that we can actually have a regular discussion and be able to access and see each other's data and share SOPs. So essentially, this is that kind of an exercise where we plan to have a similar protocol, actually the same protocol, the same kind of SOPs, the same way we're going to share and look at the data, and allowing us to actually have a cross-country tumor board on active management of these patients who have just been through molecular and genomic profiling and epigenetic profiling of their resected liver metastases. This sounds very intuitive, but we have not done this before, and we plan to do the same with imaging as well as with immunology in our setup of this Terry Fox-funded initiative. So this is just one of the workflow that we have agreed that this would work, and hopefully the reality would actually be the case that we can make this flow so smoothly between the two centers, and that we can actually make such a simple study actually work despite the complexity of the data and material transfer between the two centers. Like others, we're looking at liquid biopsy. This is we love acronyms. This is called LIBERATE. And we're behind in our group in terms of looking at liquid biopsy. What we're trying to do really is to create the framework for everybody in our institution to be able to look at liquid biopsy without having to reinvent the wheel again and again and hire different people to do the same thing. So we're actually spending a lot of time to create that flow. It will be electronic consenting. Patients just have to go to the lab, the blood will be drawn and spun and biobanked, and then it is up to the investigators how they're going to analyze the project or the samples. And if they don't have the initial idea, they can think about it and bank it and then test it later. We plan to actually start these 6 pilot projects now, looking at various tumor types to see if this will work across the institution. We're really helping them to set up the infrastructure to collect the samples. The other thing we're working with CCTG is to really go beyond the genomic sequencing. I've mentioned about the various other omics, such as full genome sequencing, transcriptomics, proteomics, immune phenotyping, epigenetics, as well as patient-derived xenografts and organoids. Obviously, this is very expensive, and none of this is really funded by any regulatory or governmental agency, so we have to fundraise and get grants for this. But this particular study that we want to start, hopefully with funding, called PROPER, is to really look at single or double or two oligometastases, patients that are undergoing surgical resection, and have a good understanding of why certain phenotype lends itself to oligometastases, and obviously all the molecular and genomic reasons behind that, versus a polymetastasis phenotype. And again, none of these projects that I've mentioned are really any more single center. We're almost always planning the next project with one or more or even multiple centers at the same time. Lee told me I have to be on time. I have 104 minutes left, so I'm going to conclude to say I think we now understand NGS as well as molecular profiling are increasingly feasible and affordable. I think we're all struck with the fact that we don't match a lot of patients, and we need to think about how to really channel the resources appropriately, including really looking at other ways to open more trials, work together to be able to actually run a study for rare variants. We need to look at combinations because clearly the success rate with single agent is just not sufficient. We're looking at functional genomics to really test the stress of therapeutics on cancers and what do they do to evolve. And obviously collective wisdom is truly what we believe in, and we've been planning protocols in every way that involve various partners around the world as well as within our own country. I want to thank all the people that are heavily involved in all these activities and obviously all the funders that make this possible, and I'm happy to take any questions. Thank you.